Action potentials in skeletal muscle cells. Do you know what's special about action potentials in skeletal muscle cells? In Part 3 of this Chalk Talk series on action potentials, we'll focus on the ion transport process in the skeletal muscle cell. The generation of action potentials in skeletal muscle cells is similar to that in neurons, which we looked at in Part 2 of this Chalk Talk series. In the default state, chloride and potassium channels are open in the skeletal muscle cell membrane, termed the sarcolemma. Together, the channels generate a resting potential of approximately -85 millivolts, which is significantly lower than that of neurons. Therefore, the sarcolemma needs to be depolarized stronger than the neuron to reach the threshold potential of -55 millivolts. Generally, this requires the accumulation of multiple stimuli at the motor end plate, termed excitatory postsynaptic potentials, in short, EPSP. The neuron releases acetylcholine, which binds to ligand-gated sodium channels in the sarcolemma. Through these channels, sodium flows down its concentration gradient into the skeletal muscle cell and the membrane potential increases. Once the threshold potential is reached, the voltage-gated sodium channels in the sarcolemma also open. Even more sodium ions flow into the cell, leading to depolarization with a membrane potential of around +25 millivolts. Similar to the neuronal action potential, the sodium channels in muscle cells begin to close before reaching the theoretical maximum potential, which we explained in Part 2. Additionally, there is delayed voltage-dependent potassium channel opening. The efflux of potassium ions down their concentration gradient repolarizes the sarcolemma. Because of the highly negative resting potential, repolarization doesn't lead to after hyperpolarization, as is the case in neurons. Whether repolarization is also shaped by the voltage-gated chloride channels of the sarcolemma, which play a role in generating the resting potential, is still subject to research. But as soon as a chloride channel opens, chloride ions flow down their concentration gradient into the skeletal muscle cell. With their negative charge, they reduce the positive excess charge in the cell, stabilizing the resting potential and reducing skeletal muscle cell excitability. Conversely, inhibiting voltage-gated chloride channels increases skeletal muscle cell excitability. Physiologically, this is important particularly when extracellular potassium levels increase as a result of muscle activity. Without a regulatory mechanism, voltage-gated sodium channels in the sarcolemma could then no longer be easily activated, leading to difficulties in forming action potentials. In skeletal muscle cells, the action potential duration is up to 10 milliseconds and slightly longer than that of a neuron, whereas the refractory period is short. For most muscles, contraction occurs only at the end of an action potential. This is because the action potential needs to pass along the sarcolemma, down the T-tubules. There, the action potential causes the sarcoplasmic reticulum to release calcium ions, initiating muscle contraction via the cross-bridge cycle. This time difference explains why skeletal muscles can enter a tetanized state, that is, they're under sustained contraction. Because of their short duration, sequential action potentials can be so close to one another that the intervals between them are about 1/3 to 1/4 of the duration of a single muscle twitch. The resulting tetanic contraction is physiologically important in generating maximum muscle strength, but can also lead to pathological muscle cramps. Such cramps need to be avoided in cardiac muscle, which we'll delve into in the next episode.